All right, so the last type of non-Mendelian practice that we're going to look at is called multiple alleles. And so really for this, what we're mainly going to focus upon is blood types. And so next class period, we will actually be doing a lab over blood types. So make sure that you are doing all of these problems. So what multiple alleles is that sometimes we have genes that have more than two alleles, like we've talked about, but an individual can only have two of those possible alleles. And so the one that we're going to look at here is human blood types. So there are three known alleles that we have. We have the allele for A, the allele for B, and then we have this recessive one, okay? A and B are codominant. So if you remember, we talked about this. Codominant means that they can both show up completely in the phenotype, okay? But they are both dominant over I, which is recessive. And next week when we're doing our lab, we'll kind of walk through like why is that possible? How does that even happen in order for that to occur? So these are all down here, all of the potential genotypes for human blood type. So if you'll notice, we have you can have two of the IA alleles, and that will give you a type A blood. You can have two IB alleles, which is type B. Two of those recessive I's, that's going to be type O. You can be type AB, which was one A allele and one IB allele. And then you can notice that for type A and B, you can also be heterozygous for both of those. Remember, like it says up here, those A and B alleles are completely dominant over the I or the recessive one. And so O and AB, you only have one option to get either one of those blood types, but type A and type B, you can be either homozygous or heterozygous. So when we look at this problem here, so we are going to cross a heterozygous person with type A blood with a heterozygous type B blood. And so similar to what we did with the sex-linked traits, okay, you want to keep the IA or the IB together, okay? So this would be our first allele, and then that second allele gets placed off above the other square, and then we have this to split up the heterozygous type B. When we fill these in, it's still the same thing as before. You're pulling the allele from both above and to the left in order to fill in those particular boxes, and these are the results that we could possibly get. And so when we look at the actual phenotypes, okay, the phenotype in this is what type of blood it is, okay? So is it type A, type AB, type B, or type O? Those are the phenotypes that we're looking at here. And so our first type is type A. And so from this particular cross, there is a 25% chance, okay? That's this genotype right here, okay? This one is going to give someone type A blood because we can see that they have that A allele here. Type B blood is going to be 25%, okay? That's this particular genotype over here because they have that B allele. Type AB is 25%, okay? When they have both of these alleles, both the A allele and the B allele, remember these are codominant, which means both are going to show up in the phenotype, and so the blood type is actually going to be AB. And then our type O possibility is 25%, and that's when we have those two recessive alleles. So go ahead and try this question real quick, and we'll see how you did. So this one says to cross a heterozygous person with type A blood with a person with type AB blood. So this is what the two parents should have looked like. So a heterozygous person with type A, that's what this first one is here, right? I've got one dominant or one allele for A, and then I have one recessive allele. And then a person with AB, we only have one option of those genotypes, and so that's what we get here. So we'll split up those alleles onto our Punnett square and then fill it in. And so as we go through this, again, when we're trying to find those phenotypes, we're really just looking at what is that blood type actually going to show. And so in this case, type A is actually 50%. When we look at these top two genotypes, even though the genotypes are not the exact same, they both are going to give us type A, okay? Whether the person has two A alleles or just one and a recessive, these are both going to give type A blood. Type B is going to be 25%, which is this one down here, the heterozygote for type B. Type AB is 25%, that's this particular genotype here. And then type O is actually going to be 0%. So there is no chance that these two people can have a child with type O blood.